Bonjour tout le monde, bienvenue à cette conférence de l'IBIS. Je vais présenter notre conférencière, puis ensuite, ça va laisser le temps aux gens de joindre à nous. Donc aujourd'hui, on a le plaisir de recevoir Dr. Kimberly Reynolds, qui est professeure adjointe à l'Université du Texas au Southwestern Medical Center. Et elle travaille dans un centre de biologie du système qui s'appelle le Green Center for Systems Biology. Kimberly a une formation en biophysique, donc elle a fait son doctorat à l'Université de Berkeley en biophysique, après avoir fait un bac à Rice University en biochimie. Donc, elle a donc une formation qui est à l'interface entre la biochimie et la biophysique. Puis ensuite, elle a fait deux stages postdoctoraux, dont un qui était avec Rama Ranganathan, qui peut-être certains d'entre vous connaissez, qui étudie l'évolution des protéines, mais comment l'évolution des protéines nous permet de nous enseigner sur la biophysique des protéines. Donc, c'est vraiment un beau lien qui fait entre les deux. C'est ce que Kim fait maintenant. Donc, elle étudie la biophysique des protéines, mais en essayant d'utiliser l'évolution pour les modifier, pour mieux comprendre comment elles fonctionnent. Donc, elle a commencé il n'y a pas si longtemps, donc en 2014, comme professeure adjointe. Et ses travaux présentement sont financés par des sources de financement prestigieuses comme le NSF Career Award. Elle a eu des fonds aussi de la Gordon and Betty Moore Foundation pour faire de la recherche qu'on appelle la Data Driven Discovery Research. Et donc, elle fait des travaux qui sont vraiment en lien à l'interface entre la biophysique, la biochimie et l'évolution. Donc, aujourd'hui, on va nous parler de comment on peut utiliser l'évolution pour modifier la régulation allostérique des enzymes. So thanks so much, Kim, for coming, and I'm looking forward to your talk.
Thank you. So thank you so much, Christian, for the invitation and for hosting me. It's really a pleasure to be able to share my work with all of you virtually. So my laboratory generally is very interested in trying to understand how evolution builds things through a process of kind of variation with selection. And so one of our goals is that hopefully by looking at many extant natural systems, which really means looking at kind of genome variation across many proteins or many genomes, can we kind of learn the principles by which evolution builds things, right? And then our hope is if we can understand what evolution and natural systems are doing, then maybe we can produce useful things in the lab. And so today I'm going to tell you about my laboratory's work in kind of engineering and understanding the optimization of allosteric regulation. I'll begin with a little bit of published work, and then there's some stuff at the end that is quite new. I was telling Christian before this, we're hoping to put it up on bioRxiv later today or maybe tomorrow morning. Okay, so as all of you know, allosteric regulation permits the control of protein function from some remote site. And here I'm showing three kind of examples of allosteric regulation. One is where you can have allosteric regulation of this active site by some small molecule ligand binding. You can have regulation through post-translational modification, like phosphorylation events, or you can even have regulation through protein-protein interactions, right? Where you have some binding event that, say, regulates ligand binding affinity over here. And in my view, all of these kind of qualify as allostery, and the key salient feature that they share is that you have some kind of remote event that modulates the protein conformational dynamics in such a way that you affect activity. Okay, so allostery is a very common feature of proteins. A lot of proteins have allosteric control. And I think one of the interesting aspects of it is that when you look across a protein family, for example, like the kinases, you can see that the allosteric regulation can occur at different sites and even by different mechanisms and different homologs, right? So here I'm showing some kinase structures. At the left is a kind of reference kinase structure, so the active site is here. And then on the right here, I'm showing these space filling representations where I've kind of painted the surfaces that serve as allosteric regulation surfaces in different kinase family members. And so as you start to color these on here, the kinase quickly starts to look like an Easter egg, because you can see that you get there are a lot of different regulatory surfaces at play, right? So some kinases are regulated by dimerization on the backside. Others are regulated by post-translational modifications in these loop regions. Still others are regulated by scaffolding modules or domain interactions here on the N-terminal lobe. And so when you look at these protein families, one question, and I think this was really nicely posed by Kurian and Eisenberg in this perspective piece in 2007, is how does all of this regulatory diversity evolve, right? And in some sense, when you think about it, allosteric regulation seems at face value a potentially kind of difficult thing to evolve, because it requires the coordination of many amino acids across some distance. And yet, when we look at families like the kinases, we see that they've evolved regulation apparently many times and at many different surfaces, right? So if allostery were truly hard to achieve, maybe we might expect to see something more like there's one allosteric site and every family member uses that site in kind of different ways. But instead we see something different, right? So how does this work? Okay, so one idea comes from actually looking at protein sequences across species. And here the kind of basic premise is that if we want to understand which amino acids in the protein are talking to each other, and speaking very loosely, right? The way that we might infer these interactions is by looking at coevolution across homologs. And so the way these analyses start is we generate a multiple sequence alignment. So we're taking sequences from hundreds to thousands of species. So the idea is these are all homologous protein sequences. And then we can analyze the amino acid conservation at individual sites, but we can also look at coevolution. So here we're looking for correlations in amino acid frequencies. So for example, every time I see a substitution at position 1, do I see a correlated substitution at position 2, right? And if I do this across all possible pairs of positions in my protein, then I can construct a covariance matrix, which is now from 1 to the number of positions in my protein. It's a square matrix. And it's symmetric, right? So the upper diagonal is the same as the lower diagonal. And each entry now tells me about the extent of correlation and, as we interpret it, coevolution between pairs of positions. And so in actuality, the data will look something like this. So here I'm showing a covariance matrix for a protein called dihydrofolate reductase. And then we can take this covariance matrix and we can analyze it using relatively standard approaches from linear algebra to define something that I'm going to refer to as a sector. And so what this sector is it's a subset of amino acids that are strongly co-evolving with each other, right? So one thing I should point out in this middle matrix is the color coding means that red is that these things are highly correlated and blue means that they're not so correlated. So you can see in this matrix, there's a lot of blue, suggesting that many of the positions are evolving relatively independently. And so the ones that I'm highlighting here are actually the ones that are in these really bright colors, which are the ones that are most strongly co-evolving in our correlation matrix. Okay, so we have this idea of sectors, and these have now been kind of analyzed in a number of different protein families. And when we look at these protein families, here I'm showing 3 examples, PDZ ligand binding domain and 2 enzymes, the serine protease and dihydrofolate reductase. Again, I'm kind of painting the sector on here in blue. What we see is that the sector itself is sparse. It's about 20 to 30% of the amino acids that are engaged in this kind of coevolutionary relationship. It's physically contiguous on the structure, right? So importantly, we didn't put any structural information into the analysis. It's just based on sequence, but we find that these form these kind of physically contiguous networks. And it connects up the active site to these distant surface sites, right? So particularly in DH-bar, you can see these kind of like little arms coming out from the active site to these distant surfaces. And so this observation it kind of suggested an idea, which is, if it's true that these co-evolving residues are really positions that are coupled to each other in some way, right? Perhaps what this represents is maybe the sector is a way to kind of wire up multiple protein surface sites to the active site, right? So here I'm showing, again, this enzyme dihydrofolate reductase, the active site's here. And so the thought is, like, if I see that this position here on the surface is co-evolving with the active site, maybe it means that if I poke here, I can actually have some kind of effect on the activity of my enzyme. Whereas if I poke at one of these gray surface sites, potentially that effect is kind of lost, or at least not communicated to the activity of the protein, right? Okay, so as a postdoc, I set out to kind of test this idea through a domain and insertion experiment, right? So the idea was that we were going to take some kind of input domain, right, that's going to sense some external signal. And then we're going to make a chimeric fusion to some output domain that we can now read out the output. And then the idea is if this becomes allosteric, then the input of this domain should now control or regulate the output of the other domain, right? And in practice, the way that this was implemented was that I took LOVE2, which is a blue light sensing domain from plants, right? And we're going to attach it into this metabolic enzyme, dihydrofolate reductase, or DHFR. And the idea is that if we're going to attach them like this so that LOVE2 is not plugged into the sector, then I'm going to expect to get no regulation. And then if I attach it like this so that LOVE2 is now plugged into the sector, I'm going to expect to get light-based control of dihydrofolate reductase with some probability, right? And it might, kind of depend on the local geometry here, but we would expect that some of the time we're actually going to get communication between these two proteins. Okay, so in this domain insertion scan, I actually introduced LOVE2 at every single surface accessible site on DHFR and then asked, okay, where do we see light-based regulation? And so here is just a really quick summary of the result, right? So I'm showing again the sector residues, these co-evolving residues in blue. These represent sequential slices through the dihydrofolate reductase structure. And then these red positions are spots on the surface where when we inserted LOVE2, we got some light-based regulation of DHFR activity. And kind of consistent with our hypothesis, I never saw light-based regulation at a non-sector connected site, but with some probability, actually about 40% of the time, we got light-based regulation when we fused LOVE2 at a site that actually was connected to the sector, so meaning within van der Waals contact in the sector. And so we saw a statistically significant association between these light-dependent positions and the spots that actually contacted the sector, and this held true over a range of kind of cutoffs for the magnitude of light dependence that we saw. Okay, so in starting my own lab, then we wanted to know, like, can we extend this principle, right? So we've shown that with domain insertion, we can introduce new regulation, right? But could we also do something analogous with arguably a more subtle perturbation, something like potentially phosphoregulation, right? So the idea is, can we engineer in new regulation by a post-translational modification? And we wanted to consider this alongside another idea. So there was a very interesting paper from Jim Farrell's group where they proposed the idea that new phosphoregulation might evolve preferentially at kind of negatively charged amino acids, right? So the idea that I thought was really cute is that in the laboratory, a lot of times whenever we see a serine or a threonine residue and we know it's phosphorylated, we make a phosphomimic of it by just making a glutamate or aspartate mutation there. So maybe nature does this kind of trick in reverse, right? So the idea is that maybe you start with an ancestral protein that has a negative charge. Then if you acquire a mutation that loses that charge, now whenever you phosphorylate it, you put that negative charge back on and you restore the interaction and thus modulate activity. Okay, so we thought, okay, this is an interesting idea. Let's see if we can combine it with kind of the idea of these sectors. And so the work that I'm about to show you is in collaboration with David Pincus, who's now at University of Chicago, and Orna Reznikov. Okay, so our model system is in yeast. So we chose kind of a prototypical MAP kinase signaling cascade in yeast. And we're looking at the serine threonine kinase KSS1. So KSS1 actually responds to a mating factor called alpha factor. And so the idea is when we add alpha factor to these cells, they trigger this MAP kinase cascade, which phosphorylates KSS1. KSS1 then translocates the nucleus, and then we activate, well, typically it activates a transcription of a pretty wide program of genes. And we also have put a YFP reporter here, right? So when we look at our cells with the YFP reporter, here what I'm showing on the y-axis is the amount of YFP expression above the KSS1 knockout. And you can see that as we add in alpha factor, so here I'm showing 4 different doses of alpha factor, that we get an increased fluorescent response. So we get this quantitative kind of report of KSS1 activity. And then the KSS1 knockout, we don't see that. And then the nice thing is we also have another kind of output, right? So in addition to this fluorescent reporter, we can also look at shmoo formation. So whenever we add in alpha factor, the yeast in preparation for mating form this kind of shmoo-like structure here, right? So we add in alpha factor and they glow and they make this shmoo structure. Okay, so the first thing we wanted to do is to ask, okay, which surface sites actually particularly which negatively charged surface sites can have an effect on KSS1 activity, right? So we're starting with this idea from the Farrell lab that D or E positions could be sites where you evolve phosphorregulation. And we just went through and we mutated each aspartate or glutamate on the surface of KSS1 one by one to alanine, starting in like effectively an alanine scan of these. And the first thing is that we saw that in general, the mutations had a pretty negligible effect on kinase expression. So these continue to be kind of made within the cell. But then we saw that they had kind of different functional effects, right? So many of these mutations were very well tolerated. And then we saw that had a handful of mutations that were inactivating, and then we had 2 that were actually a little bit activating. So they responded kind of more sensitively to the addition of alpha factor, right? And in fact, we saw, again, a strong statistical association between the mutations that had some effect and these sector-connected sites, right? So that was nice to see. But now we want to actually introduce in regulation, right? So we wanted to ask, can we now use some of these negatively charged sites to put in a synthetic phosphoregulatory signal, right? And so here's a little diagram of what we were intending to build, or we set out to build, right? So the idea is we're going to have KSS1, which we know is regulated by alpha factor, but now we also want it to talk to PKA. So we've got protein kinase A here, and it's downstream of RAS. So we've got a constitutive RAS mutant, G19B. And so when we add an estradiol, we upregulate RAS, we upregulate PKA. And then what we're going to do is we're going to drop in a PKA phosphorylation site onto KSS1, right? So we're going to introduce this motif that PKA is known to phosphorylate, which is arginine, arginine X, can be any amino acid, and then a serine residue here, right? So the idea is if everything works, then PKA should come in, phosphorylate KSS1, and regulate its activity. And so just kind of an overview of the strategy, we're going to start with a sector-connected and negatively charged residue. We're going to introduce this PKA motif, so the RRX, and the idea is we leave the charge there, provided the PKA motif itself doesn't break something, right? Then we should still have an active kinase. We're going to remove the negative charge. So we're going to put in an alanine there. And then in this case, this should now become inactive. And then now the idea is if we put a serine there, we should get something that has PKA dependent activity. So when we turn on PKA, it should phosphorylate it, restore the negative charge, and we should get an active kinase. And in the absence of PKA activity, we should have a less active or inactive KSS1. Okay, so based on our aspartate and glutamate mutagenesis results. We chose 3 sites to target for this strategy, position 8, position 68, and position 70. They're here on the kinase structure. So you can see one is kind of at the top of this N-terminal lobe, and the other 2 sit on kind of the back side of the enzyme behind the active site. And then we started to introduce these pKa sites. So at position 68, kind of the first result is that the introduction of the pKa site is just not tolerated. So clearly this position is functionally important, but this isn't really what we wanted, because now we have a kinase which is expressed, but the activation loop no longer gets phosphorylated and we don't have any signaling. Okay. But then at positions 8 and position 70, we now see something different. Okay, so on the left here, I'm showing the data for position 8, and on the right, I'm showing the data for position 70. For reference, this is the wild type response to alpha factor. And then we have these 3 different kind of substitutions at our pKa motif, right? So we can have arginine, arginine X, and then we either leave the aspartate, we put in the neutral mutation, or we put in the serine. Right? So in the absence of estradiol, right, so pKa activity is turned down, we see that this negative charge still signals roughly like the wild type, maybe with some decreased activity, but the alanine and the serine don't respond to alpha factor. And then now we're going to add the estradiol back in. And what we see is that now the serine mutant specifically can now respond to alpha factor. So now we get an alpha factor response that's conditional on the activity of pKa in our system. And so for our serine mutation, we observe pKa dependent KSS1 activity as desired, suggesting that we've managed to rewire this. And then the kind of cool thing was, you know, it's nice to see this for a fluorescent reporter, but can we actually use this regulatory system to really drive a kind of synthetic phenotypic response? And the answer is yes. So we also get pKa dependent SHMooth formation. So for the serine mutant in particular, now whenever we upregulate pKa, we also get a dramatic increase in SHMooth formation for these 2 mutants. Okay, so the idea is like, now we've shown in these 2 cases with DHFR in the domain insertion, and then now with the kinases on these negatively charged sites, that we can use kind of these sector connected surface sites to engineer in non-native regulation, right? And so, you know, we kind of proposed the idea that proteins have these conserved kind of cooperative architecture, which, you know, you can identify through this coevolutionary analysis. So we see this sector kind of embedded inside of the protein. And then perhaps what that does is it gives rise to these latent allosteric hotspots or sites with allosteric potential on the protein surface, right? And so when I look across a protein family, like the kinases, I might see that I have all of these different regulatory mechanisms, and the regulation might appear kind of very, very idiosyncratic, but maybe the thing that's enabling all of this idiosyncratic and varied regulation is actually something kind of conserved under the hood, this kind of shared sector architecture of the family, right? And so the idea is like, potentially this would really kind of facilitate rapidly rewiring or diversifying signaling systems, but it might also pose a liability, right? So potentially now you could get sort of new regulation that might not be advantageous or even disadvantageous. Okay, so, oh yes, and in a few different kind of natural and engineered systems, this principle, it seems roughly to hold, right? So here I'm showing a few different examples. This is a par6 PDZ domain. So red is the ligand binding site. This is allosterically modulated by this protein-protein interaction here. And then here we have CDC42. And you can see that when you plot these 2 proteins next to each other, that the sectors seem to kind of connect up to the interface, suggesting that maybe that's what's mediating this allosteric signal between domains. The same thing is also true in HSP70. We see a single sector that seems to connect up this ATP binding site that regulates the activity of the substrate binding site over here. And then we've also seen this now in a handful of engineered systems, so DHFR, which I told you about, and then more recently, another group engineered allosteric cathepsin inhibitor by using sector surface sites to kind of locate potential allosteric sites. Okay, so we're proposing that these sectors give kind of a substrate for evolving allosteric regulation at these conserved surface sites. But then what happens next, right? So one of the things that I didn't tell you in our earlier work was that many of these kind of initial allosteric fusions, particularly for the DHFR LOVE2 systems, they show detectable allosteric regulation, but that allosteric regulation is really quite modest. So we might see that the activity of our output enzyme DHFR is modulated by 15 or 20% in response to light or input, right? This is hardly the kind of like fold effect size that biochemists often characterize in natural regulatory systems. So, once you initiate allostery, whether through kind of engineering or some evolutionary event like a domain recombination, you want to understand how and where is it optimized, right? So specifically, like, is the pattern of mutations that can influence allostery sparse, right? Is it just a few mutations, or is it like almost all of the mutations? Is it abundant? Secondly, is the pattern of mutations that can influence allostery really localized? Like, is it focused around, say, this domain insertion site, or is it really distributed throughout the protein? And then finally, is there any kind of relationship between the mutations that influence allostery and the sector, right? Okay, so this is relatively new work that I'm going to tell you all about. And it's really being led by a really great postdoc in my group, James McCormick. And then it was assisted by Samuel Thompson, who was a graduate student in Tanya Corten's lab. He made our deep mutational scanning libraries, actually. And then Max Russo, who's a research assistant in the lab that did a lot of kind of in vitro biochemistry work that I'll show kind of later. Okay, so we want to understand how these allosteric systems get optimized or tuned up. And so our starting place for this is going to be one of these DHFR LOVE2 fusions that I mentioned kind of at the very beginning of the talk, is a kind of initial system for characterizing whether or not these sector connected edges were hotspots for regulation. Okay, so here we have DHFR, our output enzyme. And then here I'm showing the LOVE2 domain. And the construct that we're starting with, we call it DL121 for DHFR LOVE2 121, because LOVE2 is inserted between positions 120 and 121 on the DHFR backbone. And one of the convenient things from an engineering perspective about LOVE2 is that the N and C termini are really close together. So the fusion is literally a chimeric construct. We're inserting LOVE2 into the backbone of DHFR covalently. Okay, and then LOVE2 in its kind of natural setting is thought to communicate like the input of light through this J alpha helix here that's colored in red. So basically light is absorbed at this FMN chromophore, and then in response to light activation, that helix is thought to kind of unravel. So there's a part unfolding of the protein. And then somehow, we don't know how, right? This is communicated to our DHFR domain where it's inserted in this loop, which is in this loop region that's known to change conformations during the catalytic cycle, right? So there's some hint that this site is important to catalysis already. Okay, and this starting construct has kind of a modest allosteric effect. So we get about 20% increase or 28% increase in the Kcat, so the enzymatic velocity in response to light. So here are the in vitro measurements showing that. And the other thing that's nice about this system is that dihydrofolate reductase, because it's an essential metabolic enzyme, it's fairly straightforward to link the activity of DHFR to the growth rate of your cells, right? And so the idea is that we can use growth rate as kind of a proxy for the catalytic activity of DHFR. And so what we can do is we can take cells where we've transformed this DL121 construct. We can grow them in the light, we can grow them in the dark, we take the growth rate difference, and that tells us if we can see an allosteric effect, right? So here I'm showing, and we'll come back to this assay in a moment, but here I'm showing data for a few different point mutants in a non-chimeric DHFR with no LOVE2 domain. These show very minimal growth rate differences in the light versus dark. And then DL121, we get a 20% increase in the growth rate in the light versus the dark. Okay, so the idea is we're going to start off with this system where the input signal of light is very rapidly applied. So we can put light on or we can take it off. The allosteric effects on activity, we can either quantify with these in vitro assays or with much higher throughput by looking at in vivo growth rates. And we expect that there's kind of large improvements in allosteric dynamic range. There's a potential for that, right? Because we're starting from kind of a very modest allosteric switch. Okay, so here's our experiment. What we're going to do is do a deep mutational scan of our DHFR domain in this fusion under lit and dark conditions, right? So we're going to make every mutation to all possible amino acids at all 159 positions of DHFR. And then we're going to measure the effects of those mutations on growth rate in the light. We're going to measure the effect of that mutation on growth rate in the dark, right? And then we're going to subtract these two to infer whether or not there's some allosteric effect, right? And so we can do that with high throughput for this full mutational library. And so doing this, like going into this, we expected that many of these mutations would potentially have relatively small effects. So we wanted to make sure that the resolving power of our assay was as kind of excellent as possible, because we didn't want to sort of be missing mutations because they were within the error of our assay, right? So I told you that we can see, or we can use growth rate as a proxy for activity. And so here what we did was to really optimize our assay so that we could have that relationship to be as kind of as quantitative as possible, right? So the first thing that we do is we do our entire assay in a turbidostat. So basically we take our deep mutational scanning library, we transform it into a population of E. coli, and then we're going to grow them in a turbidostat. And what this is a device for continuous culture where we can continually put in fresh media so that they stay clamped at a fixed optical density, right? And then we have a waistline, so we're effluxing out the waste so our tubes don't overflow, right? And then our turbidostat is equipped with blue light so that we can have some of the vials on and some of the vials off. And just to show like what the data for the turbidostat looks like, is that now basically what you see is these little teeny cycles of growth and dilution where the cells grow and then we dilute in fresh media and then they grow and then we dilute in fresh media. And so the idea is we can keep them under kind of stable media conditions and growth state conditions for the duration of our experiment, which really lasts about 24 hours. Okay. So now we're going to take our library, we're growing it in the turbidostat, and then we're going to take out time points, and we're going to deep sequence to look at allele frequencies over time. And just so to start off with, to kind of calibrate our assay, we're beginning with a set of wild type DHFR, so it's not fused to LOVE2. We have DL121, our fused construct of interest, and then 11 point mutants of DHFR. And these are all point mutants of DHFR that we've previously characterized the catalytic activity for in the lab, right? And so we've got these all growing as a mixed population, and then we're sequencing basically the frequency of each of these alleles at every time point, right? So for very, I guess, unfit alleles, we see that they decrease in frequency over time. Everything in our experiment is normalized here to the wild type and then normalized to T0, right? And we're taking a log. So these things are exponentially growing, right? So that's why we need a logarithm here to get this linear relationship. And then we can fit a slope to this. And what that slope is now the relative growth rate of each mutant relative to wild type. So it's basically saying this slope is the difference in growth rate for this variant compared to wild type. And under the conditions of our assay, so James went through and very carefully kind of tested a few different media conditions to try to make this relationship as linear as possible. We get a really nice kind of log linear relationship between the relative growth rate on the y-axis and then the catalytic activity here, or log catalytic activity on the x-axis. And so this linear relationship holds over about 4 orders of magnitude in DHFR activity. And then you can see at some point it kind of saturates, right? And having a more active DHFR doesn't really give you much. And then DL121, our mutant of interest, is kind of near the center of this regime. I'll point out that DL121 does not sit on the calibration line as nicely as the rest of the mutants. It is the one variant in this experiment that has a LOVE2 domain included. Our hypothesis is that this variant just isn't being as expressed as highly as some of these other things, and so it falls off this line. But I want to emphasize that for our allostery measurements, because we're looking at the relative growth rate in the lit versus dark, as long as changes in expression aren't somehow light dependent, those kinds of things should normalize out. Okay, and so we're going to take this assay and we're going to use it to measure relative growth rates for all of our DL121 mutants under dark and lit conditions in triplicate. Okay, so before I go into the allostery effects, first we're just going to consider the effect of mutations on growth rate in a single state, right? So this doesn't really tell us about allostery, but first we would just want to make sure that kind of our distribution of fitness effects makes sense, and that we see things like, stop codons are deleterious, and mutations that are known to be critical for DHFR function are coming up as deleterious, right? And so we can plot these data as a heat map here. With this color coding blue are deleterious mutations, red are things that are beneficial. Null data are mutations that actually dropped out or we couldn't see by sequencing after 3 time points, so we couldn't fit a slope. So we infer that these are actually some of the least fit alleles in our population. And then no data, we have a handful of mutations with no data that are in gray. These are things that were just, for whatever reason, not included in our original library construction or that we didn't see post-transformation. Okay, so if we look at the distribution of fitness effects relative to our starting construct, DL121, so this is our fusion construct sits here, we can see that the mutations are really largely deleterious, right? We see a handful of beneficial mutations kind of near the LOVE2 insertion site, but in general, this thing is not very mutationally robust. Potentially that makes sense because we know that DL121 is starting with reduced catalytic activity relative to DHFR. So it may be that it's just kind of more mutationally sensitive. And then we can look at the positions that are really enriched for deleterious mutations, right? So we're going to take all of the kind of most deleterious mutations in our library. And the way that we decided on these mutations was using as a cutoff DL121 D27N. So I should explain that the D27N mutation is a mutation in the active site of DHFR that renders it not quite, but close to catalytically inactive. So you can kind of barely measure its activity. And it sort of minimally rescues growth in our assay system. If you remove a few of the nutrients from the media, these cells are dead. So we're taking everything from here down as extremely deleterious mutations. And if we look at these mutations, they're kind of enriched in places you might expect. So around the cofactor binding site and around the active site. And they show a statistically significant kind of association with sector positions, right? So this is consistent with the view that the sector, these co-evolving residues are critical to protein function. I'll also point out that it shows a statistically significant association with conservation, right? So a lot of conserved positions are included in the sector. And that again makes sense that the most deleterious mutations are going to occur at conserved sites. Okay, and then now what we're going to do is we're going to remove the slowest growers prior to our analysis of allostery. So we're going to take everything to the left of this D27N line, and we're going to exclude them from the analysis of allostery. And there's kind of 2 lines of thinking for this. First of all, inactive mutants are by definition kind of not allosteric, right? They can't be regulated if they don't have some base catalytic activity. And so here, this is just showing that after we remove these deleterious mutations, right? The deleterious mutations are a pretty good chunk of our library. So we're left with about 51% of the overall library, so 1,548 mutations. And then once we apply that filtering, these mutations are still fairly well distributed between the surface of the protein, the core of the protein, sector positions, evolutionarily conserved positions, right? So we believe that after this filtering, we still have kind of a representative set of mutations for analysis. And the second thing that it does in applying this filtering to remove the slowest growing mutants is we're also removing some of the noisiest measurements from consideration, right? So here what I'm showing are plots of, we did 3 replicates of this experiment. So I'm showing 1 versus 2, 2 versus 3, so on. And so you can see that the slowest growers, these cyan lines are where we're drawing our cutoffs. So these are the ones that are some of the noisiest data. And so we're not going to consider those when we try to infer allostery. Okay, so now for each mutant, we've got these triplicate lit and dark measurements. And these are just showing some representative curves for 3 different point mutants in our library. We have D27N, which I already told you was very deleterious. So we can see that it decreases in frequency quite a lot, whether it's in the light or in the dark. We have DL121, D122W, which is modestly beneficial in the dark and in the light. And then we have things like E154R, which is sort of more deleterious in the dark than in the light. And these are the kind of allosteric mutations that we want to identify. Okay, so to do this statistically, what we did was to compute a p-value for each one of our mutations. And this was done by a t-test to basically ask, are the means, what is the probability that the means for the lit and the dark measurements are the same, right? So we get a p-value describing the significance of that difference, and then we're plotting that to the relative growth rate difference. This is our allosteric effect here on the x-axis. So mutations on the right are things that are allostery enhancing. Mutations on the left are things that our assay is reporting are allostery disrupting. And we can look at these with respect to 2 different p-value cutoffs. One is a standard p-value cutoff of 0.05. And the other is a more stringent p-value cutoff of 0.016. This p-value cutoff reflects kind of an adjustment for multiple hypothesis testing. So the idea is, because we have a really large number of data points, we want to correct for the fact that we're kind of doing so many statistical experiments, right? So the data I'll show today are for this adjusted and more stringent p-value, but I'll point out that the overall results also hold true when you consider this kind of standard p-value cutoff of 0.05. Okay, so and the red points that I'm highlighting here are things that we later went on and characterized in vitro. I'll talk about those more in a moment. Okay, so we look at this broadly, and in total, we found that there were 69 significant allosteric mutations, about 5% of the library. So even though most of the mutations had a deleterious effect on function, right? These are in large part not neutral mutations. Only a small proportion have significant kind of differential effects in the light versus the dark. So really a sparse subset seem to tune allostery. We found that 56 of them actually enhance allostery, and then 13 are allostery disrupting. And here I'm just showing the growth rate data. So here are the 3 lit replicates and then the 3 dark replicates for each of these significant mutations. So you can get a sense of the data a little bit more. And importantly, we didn't really see a strong association between the allosteric effect size and growth rate, right? So we find some mutations which are growth rate enhancing benefit allostery, but then we also see growth rate, allostery enhancing mutations at these lower growth rates as well. Okay, and then as a check, we wanted to ask, is our relative growth rate difference really reporting on our kind of in vitro measures of allostery? So we went and expressed and purified and catalytically characterized a handful of mutations with kind of a range of allosteric effects. And we see that we get a pretty good relationship between the in vitro allosteric effect, so the ratio of the K-cat and the light to the dark, and then the relative growth rate difference in our assay. So indicating that our assay really is kind of reporting on the changes in allostery for these point mutants. And the mutation effects that we're getting are fairly modest. Perhaps that's what you might expect for single mutants. But we see, for example, like the strongest mutation that we characterize in vitro, H124Q, increases regulation to twofold. So now we see a twofold effect when we hit this thing with light in terms of how upregulated the catalytic activity is, right? And just as a reminder, our starting construct, DL121, shows a 28% increase. So this gives us a pretty good bump. Okay, so where are these mutations on the structure? All right, so the allostery disrupting mutations, we have, what did I say, 13 of those. They localize to 6 structural positions, most of which are right here around the LOVE2 insertion site. So LOVE2 is inserted between 120 and 121. And if we plot kind of the fraction of allostery disrupting mutations as a function of the distance to 121, it's just really clear, right, that 90% of our mutations are occurring within 10 angstroms of that insertion site. And so it kind of suggests that maybe these mutations are kind of disrupting some local communication or structural interactions with LOVE2. I think it kind of remains to be determined, but they're all very proximal to the insertion site. Now for the allostery enhancing mutations, we saw something that frankly we were quite surprised by, which is that the allostery enhancing mutations are distributed across the protein surface. So they're not proximal to the insertion site. They're just sort of all over the surface of DHFR. So we have, I think, 56 enhancing mutations, and they occur at something like 25-ish positions. And we see kind of a statistically significant association between these allostery enhancing positions and solvent accessible positions. So many of these are located on the protein surface. And here I'm just going to show you the data in relationship to the sector. So again, I have the same color coding. These light blue positions are the positions that are allostery enhancing. The orange ones are the positions that are allostery disrupting. And again, the dark blue is the sector, right? So you can see the disrupting ones are all here, or mostly, except for one, kind of around this LOVE2 insertion site. And then we get these enhancing ones, some of which are contacting the sector, but some of which are occurring in these kind of loop regions, which don't even really make contact with the sector. Many of these are occurring at fairly weakly conserved sites. And in fact, when we look at the association of these allostery enhancing mutations with the sector, we find that none of them are directly occurring within the sector, right? And so, in fact, we actually see kind of depletion of allostery enhancing mutations in the sector. And we see a modest enrichment of allostery disrupting mutations in the sector, right? So this was really interesting to us because it suggested, like, if we make a mutation in the sector, either it's going to be really deleterious to function or it might disrupt allostery. And then if we want to benefit allostery or tune up allostery, what we want to do is instead kind of leave the sector alone, but fiddle around with these surface residues that are occurring in these more kind of weakly conserved regions, and in some cases in like loop regions on the surface of DHFR. Okay, so to kind of summarize this study, what we found is that these sector-connected surfaces are hotspots for putting in new allostery, that mutations that disrupt function are enriched in the sector, and while single mutations can exert a large effect on function, the kind of effects of these single mutations on allostery are comparatively modest. And in fact, really only a small fraction of the mutations, about 5%, we observed even modulated allosteric regulation. We saw that these allostery disrupting mutations seem to be localized to the LOVE2 insertion site, and that the allostery enhancing mutations are distributed across the protein surface. So I just want to leave you with a kind of couple of like last experiment that we're pushing forward on, which is, okay, we see that these surface sites can enhance, or surface mutations can enhance allostery, by like say twofold, right? So can we actually start combining these to generate something more like a high performance switch, right? So we started doing this, right? So here we're going to consider 3 mutations, M16A, D87A, and H124Q. Individually, these all enhance allostery relative to our starting DL121 construct. This is the fold effect here in white on these bars. And then we can start combining these. And one thing that we're kind of encouraged by is that the observed allosteric effect of the combinations is actually really close to what you would expect if you were going to just take a log additive model and say, like, okay, I'm just going to take a log sum over these fold effects, right? So the observed is actually quite close to the expected, suggesting that you can maybe combine these in a predictable way. I mean, I don't know, we'll have to see. And so kind of our idea is that maybe you could get new regulation by having some kind of domain insertion event, right? That fuses now this domain to a sector connected surface. And then through iterative surface residue variation, you can now start to tune this relationship up so that you can start to get a stronger communication between these two domains. And so with that, I'll conclude and thank the people who really did the work. So the study on engineering kinase regulation that I showed you at the beginning was done in collaboration with David Pincus, his postdoc Jay Prandy, and Orna Reznikov. And then the DL121 saturation mutagenesis and optimization work was done by James, Samuel, and Max. And then of course, thank our funding. And I'll point out my lab is recruiting, where I'm actually actively looking for a postdoctoral fellow to recruit to kind of our studies of optimizing and engineering allosteric regulation. We're located in Dallas, Texas, and here's a picture of our campus. Yeah, we'll welcome any questions.
Thank you very much, Kim, for this brilliant talk. It was very interesting. Do you have any question for Kim? Avez-vous des questions pour Kim? Si oui, vous pouvez allumer votre micro puis poser votre question. Maybe I can start while people warm up.
Yeah.
So you thanks a lot. That was awesome. I have a few questions, like more general questions, and one more specific. In the latest part where you divide the sites as being allostery disrupting or enhancing, do you have sites that are both, but that depending on the residue that you put there, or is it always like one or the other? It doesn't depend on the exact substitution, amino acid substitution that you do.
Yeah, that's a great question. So we have, and most sites are either one or the other. There's a couple of sites, let's see, kind of directly near the insertion site. There's maybe one or two positions here where, depending on the mutation that you introduce, it could be enhancing or it could be disrupting.
Okay.
Yeah,
cool.
so I think there's probably two places where you can see either.
Is that what you would have predicted, that the two sets of sites would be almost exclusive?
No.
Okay. Because my intuitively I would say it would be the same site, but depending on what you put there with enhance or disrupt allostery, I would not have predicted that.
Yeah, I agree. So our expectation was that we were going to see a lot of things around the insertion site, and then potentially we might see some mutations kind of within the sector. We also thought that it was going to be kind of one set of positions where the identity mattered. But yeah, we saw something that indicates kind of a different model, right? And it seems like the sector itself, like you just don't want to, like, I guess you don't want to mess with it, right? I'm speaking kind of loosely, but the mutations there tend to break things more than give you kind of allostery enhancements. One experiment we've been kind of thinking about is, we're starting from this initial kind of naive fusion where the DHFR activity is already knocked down, right? So like, what would happen if you repeated this study in the background of like a mutation that's stabilizing or activity enhancing that kind of bumps it back up? Would we see a different pattern?
Yep.
Yeah.
Yeah. Awesome. Thanks.
Mm-hmm.
Est-ce que vous avez des questions?
Maybe I have a question on maybe the part on the phosphorylation that you showed us. So you presented like the really nice model and process by which you try to add the synthetic phosphorylation site. Do you think it would be possible to try and use like a multiple sequence alignment to detect something like that happening during the course of evolution? Because in theory, like it it might be possible to see it if you compare many proteins of the same family.
Yeah, I think that would be a really interesting follow-up. It's something, yeah, it's something we haven't done. I will kind of point out that in the original study from Jim Farrell, where they proposed this idea, the way they kind of checked their idea was to make multiple sequence alignments and phylogenetic trees for proteins with known phosphorylation sites, and that they showed that you know, more often than one would expect, the ancestral state of that was a negatively charged amino acid, right? So yeah, I think it would be kind of interesting to now add like the sector connection as another dimension to that, to ask if that can kind of refine your hypothesis of where those sites might evolve. But yeah, it's something we haven't done yet, but I think would be interesting to try.
And maybe a follow-up also on the phosphorylation stuff. Do you think you could use kind of this approach of on the sectors and the phosphorylation side to try and identify variants that have effect on phenotype? Because it's very difficult to know if phosphorylation has actually an effect on protein function just computationally. So do you think that would be kind of a better approach to that problem?
Yeah, I think that would also be a really interesting thing to try, which we haven't done yet. And you know, I was talking to my collaborator, like David and Orna have done a lot of kind of mass spec work, and we were talking about the fact that a lot of times by mass spec, you can see that there's a lot of phosphorylation, but it's not clear which of those sites are actually functional or have any kind of impact, right? So we started kicking around the idea that maybe you could use sectors to try to prioritize things that have a functional effect. And I think really the limitation the last time we had talked about it was having a good gold standard data set of like phosphosites where you could say these are functional and these are not, where we could really test it and say like, is it working, right? But I think if we had good gold standard data, it would be a nice thing to try for sure.
Other questions?
Yeah, I think I have a quick one. Yeah.
Go ahead.
So I was thinking about the allostery enhancing mutations, and I was wondering if maybe some of those positions would have been found to be like co-evolving with each other. Like say maybe they form part of another sector that's also co-evolving or something like that.
Oh, yeah. I mean, I think we could definitely go back and look at those positions more carefully now that we know which ones they are. I mean, I think one of the kind of interesting things to me is that they're sort of weakly conserved. So in general, they're not something that that pops up into our sort of our usual sequence analyses because they vary so much. We don't really see a lot of statistical signal for them. Yeah.
Okay, thanks.
Mm-hmm.
Any other questions for Kim?
I have one question which quite closely follows on from Angel's question. So when you were talking about mutations that enhance allostery, you found that many of them actually map to different like loops within the protein structure. And I was wondering if you've ever thought about the fact that in effect, these loops may actually technically be part of a sector in effect, but you just can't detect them because of problems with the alignment. So with a lot of these loop sequences, it might be very difficult to align them, and your definition of a sector is based upon the alignment. But in effect, these loops might be very physically flexible. So the actual loop itself may form a part of a sector, but no one individual position in that loop, or amino acid in that loop, may actually be detectable as a sector residue.
Yeah, I think this is a really interesting comment, and something that we've been thinking about, but I don't have a good answer for at present. Is I think, as you see, it's very possible that maybe these are part of the sector and we don't detect them either because the loop regions are maybe less reliably aligned, or you know, maybe it's possible that in some conformations, like these loops do kind of contact or influence the sector positions, and you know, we just don't see that in kind of our static structure that we have here. So yeah, I think, we're still kind of trying to think about how to get at that, and also like just even the basic mechanism of how some of these mutations are working. So some things that we're thinking about is to try to get, well, first of all, we don't have a structure of the DL121 fusion. So can we get a structure of the fusion and then maybe crystallize a few of these mutants? We're also thinking of doing, if we can have a good structure or even a good model as a starting place, could we do some MD simulations to try to see like how these mutations might be influencing dynamics, or are they somehow propagating through the sector, or is there some other mechanism that's happening? And that right now, I think we just don't know, but it's very interesting to me that they're all over the surface, right? And like, how is that even working? Yeah.
Yeah, I think that's very interesting. Thank you.
Thank you.
All right, thank you very much. I think we'll stop here for the general questions, and Kim will stay with us for maybe an hour to discuss with people. So thank you very much, Kim. I'll let people go, and it was a great talk. And if you want, we'll leave you maybe 10 minutes if you want to take a break, and we'll reconvene here for the meeting.
Okay, sounds great. I will be back in 10
Awesome.
minutes. Yeah,
Thanks a lot.
Thanks.